Living things produce a waste product called hydrogen peroxide. It's a very nasty substance. To break it down, they use the enzyme catalase. Today, as our source of catalase, we'll use celery. And to extract the catalase, we first of all chop the celery. Using a chopping board to protect the bench. And we'll transfer the chopped celery into a blender. So the enzyme is catalase, and the source of our enzyme is celery. We'll add some liquid just to allow it to blend. And we blend it for 4 seconds. If we blend it for any longer, we'll chop up the enzyme and destroy it. So 4 seconds. To extract out the enzyme and to separate out all the cell parts. In here now we've got celery, all the cell walls, cell membranes and stuff like that. We don't want it. We filter it using coffee filter paper. Coffee filter paper allows it to filter very quickly. Lab filter paper would be too slow. So we just pour the blend into the filter paper, and you can see the filtrate coming through. So we just wait for a sufficient amount of filtrate to come through. Having allowed the filtrate to pass through, we transfer it to a beaker. It's easier to get at. And we'll use the syringe to measure out a certain amount of the filtrate, which contains the enzyme catalase. This time we're going to use pH 10 for all the experiments, because catalase, we found in a previous experiment, catalase works best at pH 10. Its optimum pH is pH 10, so we're going to use pH 10. We transfer 10 cubic centimetres of the blended celery in here. We take 10 cubic centimetres of pH buffer 10. And to create the foam, we use 2 drops of washing up liquid. The substrate is hydrogen peroxide, which is harmful, so using gloves to protect the hands. And we transfer 10 cubic centimetres of this into, 5 cubic centimetres of this into a graduated cylinder. Now, we want to bring the 2 of these to a temperature of 0. To get the water bath down to 0 degrees, we use ice, and we use an ice bath. So we break some ice in here and transfer it in. And that will bring the water bath to a temperature of 0. Having got the water bath down to a temperature of 0, we put our 2 graduated cylinders in here to bring the 2 of them down to a temperature of 0. We leave them there for 10 minutes. After 10 minutes, we take both graduated cylinders out of the water bath at 0 degrees, and we add the hydrogen peroxide, which is the substrate, to the enzyme, which is the celery solution. And we see that at 0 degrees centigrade, no foam is produced. The enzyme is inactive at 0 degrees centigrade. We repeat the experiment at 10-degree increases. In other words, at 0, 10, 20, 30, 40, 50, and 60 degrees. This is one that we've had in here for 10 minutes at 30 degrees centigrade. So we take it out, and we pour the hydrogen peroxide into the celery solution, which contains the enzyme. And straight away, we see that there's a foam appears. We wait 2 minutes to see what height the foam reaches or what volume of foam we get. In doing the calculation, you have to subtract the volume of liquid at the bottom, which is 25 cubic centimetres, from the volume of the foam at the top to get the right answer. Here we have the results of the experiment. At 0 degrees centigrade, no foam was produced. The enzyme was inactive at 0. At 10 degrees centigrade, a small volume of foam was produced. At 20 degrees centigrade, a reasonable amount of foam was produced. At 30 degrees centigrade, slightly more foam was produced, making somewhere between 20 and 30 the ideal temperature for catalase. At 40 degrees centigrade, less foam was produced. And at 50 degrees, hardly any foam was produced. That shows the enzyme is becoming inactive. And at 60 degrees centigrade, no foam was produced. The enzyme was inactive, probably denatured.